Okay, so remember we said that last time that pyruvate, which is a 3-carbon molecule, is going to be converted to acetyl-CoA, which is a 2-carbon molecule. The extra carbon is going to be lost in the form of carbon dioxide, and NADH is produced. So the pathway of glycolysis runs in the cytosol, while the TCA cycle, the next part, occurs in the mitochondria. Basically, pyruvate has to travel from the cytosol to the mitochondria for this entire pathway to occur. So all of the enzymes and precursors are going to be found in the matrix of the mitochondria. And remember, the matrix is the fluid inside the mitochondria. So this pathway is really essential to metabolism because it's going to accept and give other metabolites to other pathways. So for example, right here we see glutamate to protein synthesis. So the TCA cycle also produces some NADH, which is a reducing molecule, which eventually makes ATP. So right here we can see that pyruvate can make lactate, it can make alanine, it can make oxaloacetate, and it can also make acetyl-CoA. Acetyl-CoA right here and oxaloacetate are both part of the TCA cycle. For pyruvate, to make pyruvate into acetyl-CoA, you produce a carbon dioxide molecule as well as one NADH. And to make oxaloacetate from pyruvate, we're going to use one carbon dioxide molecule and we're going to use one NADH. So basically they're the complete opposites of each other. Pyruvate to acetyl-CoA produces one carbon dioxide and one NADH, and then oxaloacetate is going to use the carbon dioxide and one NADH. So pyruvate dehydrogenase, or PDH, catalyzes pyruvate into acetyl-CoA, and this means that it's going to be taking away a carbon and releasing it in the form of carbon dioxide. While pyruvate carboxylase, this thing right here, is going to attach a carbon onto pyruvate to make oxaloacetate, and that's why we have to utilize the carbon dioxide to go from pyruvate to oxaloacetate. So typically the TCA cycle does not use the pathway from pyruvate to oxaloacetate, and it's actually going to be the pyruvate to acetyl-CoA pathway that is more favored for the TCA cycle. So PDH, or pyruvate dehydrogenase, is a very complex enzyme. It's going to have a lot of cofactors, one of them being vitamin B1. And remember that vitamin B1 deficiency causes beriberi. So in the absence of vitamin B1 or any of the other cofactors, this is going to cause the enzyme activity, this enzyme, to decrease, and therefore it's going to cause the production of acetyl-CoA to decrease. So if there's not enough acetyl-CoA, then there won't be enough energy in the body. So here's another picture of the TCA cycle. The first step right here is the condensing of acetyl-CoA and oxaloacetate. So the conversion of pyruvate to acetyl-CoA is not part of the TCA cycle. That's actually going to happen after glycolysis and before the TCA cycle. So as you can see in this pathway, one acetyl-CoA molecule is going to make 1, 2, and 3 NADH, and it's also going to produce 1 FADH2. But keep in mind that glycolysis makes 2 pyruvate, and therefore 2 acetyl-CoA molecules will enter the TCA cycle, doubling the product. So in the end, we're actually going to have 6 NADH and 2 FADH2. Also, keep in mind that 2 GTP are going to produce, and GTP is a lot like ATP. Also, just take note that whenever NADH or FADH is formed, the enzyme that is used is a decarboxylase. It's going to be a dehydrogenase. You can see the enzymes right here. And this makes a lot of sense because a dehydrogenase is going to take away a proton or a hydrogen molecule, and as we can see right here, the results are NADH and then the FADH, and they come from something like NAD, which does not have a hydrogen, so that hydrogen that the dehydrogenase is taking away is just going to end up on the product NADH and FADH2. So if there is low oxaloacetate, then the TCA cycle is going to slow down, and it causes acetyl-CoA to be converted to oxaloacetate through carboxylation, or adding a carbon dioxide. So the reaction that converts acetyl-CoA back to oxaloacetate is called an anaprolytic reaction, and this is just going to describe the deficiency of oxaloacetate in the beginning. So this chart on the left side shows the net gain of products in each process of glycolysis, pyruvate to acetyl-CoA, and then the TCA cycle. The right side right here is going to show how many molecules of ATP can be formed by each product. So we see that for glycolysis and for pyruvate to acetyl-CoA, that 2 NADHs are either going to produce 4 ATP or 6 ATP. So this doesn't really make any sense, except that we have to consider that glycolysis is going to take place in the cytosol, while the conversion of pyruvate to acetyl-CoA is taking place in the mitochondria. So in summary, NADH to ATP is 2 ATP per NADH in the cytosol, but then in the mitochondria it's going to be 3 ATP molecules per 1 NADH. Also, just take note that FADH2 produces 2 ATP molecules. So you see that right here. And the reason why NADH only produces 2 ATP molecules is because in the cytosol is because it's actually going to follow NADH2 in the cytosol. So one of the regulatory enzymes of the TCA cycle is PDH, or pyruvate dehydrogenase. And PDH is a complex of many enzymes that goes back and forth from being active to inactive. So high amounts of NADH and acetyl-CoA are going to stimulate the PDH kinase, which is going to phosphorylate PDH, inactivating this enzyme. So PDH phosphatase right here is what's going to dephosphorylate PDH and making it active again. But first, PDH is going to have to be activated, and the way it's activated is by NAD+. And NAD+ is just NADH after NADH has been utilized. So active PDH will keep the TCA cycle going. So this whole thing makes a lot of sense because PDH phosphatase is only active when it's needed, and it is active when NADH has been used and there needs to be more NADH produced. So it gets activated and then used, and then it uses up and then it's activated by the used up NADH, and it keeps the TCA cycle going. So in this picture, we see ATP being formed in the mitochondria by NADH and FADH2. For every proton, one ATP is formed. So there are 3 protons formed by NADH, so 3 ATP produced. And then FADH2 makes 2 protons, so 2 ATP are produced. This next picture just shows us how NADH produces 3 protons and FADH2 produces 2 protons. So NADH right here is going to go down this whole pathway through 1, 2, 3 complexes, which allow electrons to flow through with NADH 3 times, making 3 protons. So FADH is actually going to bypass this first complex right here, and it's going to start with ubiquinone. It's going to go down 2 complexes with electrons and produce 2 protons. So going back to this picture, we can see that ATP synthase right here is located on the inner membrane of the mitochondria. This enzyme is going to produce 1 ATP for every proton that passes through it. So most of the ATP made in the mitochondria is actually going to be used inside, so ATP has to be transported out of the mitochondria by the ATP ADP translocase right here. This is an antiporter that moves ADP inside and ATP outside. The ADP that goes back inside the mitochondria is going to be recycled back right over here into ATP, and then is going to be transported out again of the mitochondria by the ATP ADP translocase. So here's this pathway again. There's an insecticide called rolinone right here, and this is going to inhibit complex 1, meaning that NADH is not going to be able to give an electron away, and the production of protons stops right here and kills the cell. But FADH actually can still proceed to make protons because it doesn't go through complex 1. The only problem is that the production is going to be super limited, and the cell gets most of the energy from NADH anyway, so this wouldn't really matter. So antimycin right here, A, inhibits complex 3, which inhibits both the FADH and NADH from making protons. But antimycin A can be overcome by vitamin C, and vitamin C is going to help accept protons and restore respiration. So let's talk about carbon monoxide. So hemoglobin has an affinity for both carbon monoxide as well as oxygen, but hemoglobin has an affinity for carbon monoxide almost 1,000 times that of oxygen. So even if there's a tiny amount of carbon monoxide, it will saturate hemoglobin, making it so that it can't carry oxygen. So carbon monoxide also inhibits complex 4 right here, and if that happens, there won't be any electron flow, thus no proton production, and this can be overcome by methylene blue. This will accept electrons from complex 3, and then the electron flow will continue making protons. So as we already know, maltose is made of glucose and another glucose because it's a disaccharide. Sucrose is glucose and fructose, and then lactose is glucose and galactose. So right here, fructose is going to be converted to fructose 1-phosphate by fructokinase. And remember, a kinase is just going to add a phosphor group onto the fructose. Then fructose 1-phosphate is going to be cleaved by fructose 1-aldolase, and we're going to get glycerol plus DHAP. So glycerol will then be phosphorylated to make glucose 6-phosphate, and this is how fructose enters the glycolytic pathway. So now for galactose. Galactose is going to be phosphorylated by galactokinase, making galactose 1-phosphate, and then in the presence of a UDP glucose, which is the active form of glucose, galactose 1 phosphate is going to be converted to UDP galactose and glucose 1 phosphate. So they're basically going to switch places. And then what's going to happen to glucose 1 phosphate is that a phosphoglucomutase is going to move the phosphate on carbon 1 to carbon 6, and this is going to make glucose 6 phosphate. And we all know this is the first molecule that starts. That's at the start of glycolysis. Right, looks like that's all for today. See you guys next time.